Elearning Jamaica Limited, in association with the Ministry of Education and Czec Jamaica Limited, present a series of e-learning videos geared towards preparing students for CXC examinations. This series focuses on biology and highlights areas considered most challenging to our students. There are 10 videos, each lasting 20 minutes. Your narrator, Michael Anthony Cuff.
Learning outcomes. After completing this lesson, student will be able to outline the process of meiosis. There are 46 chromosomes in each human cell except red blood cells and gametes. In gametes, sex cells, there are 23 chromosomes. Can you imagine what would result if gametes contained 46 chromosomes? As a result of fertilization, the resulting offspring would have 98 chromosomes in each cell. Furthermore, the number would keep on doubling with each successive generation. For this reason, a process by which the chromosome number is halved and chromosomes are sorted and packaged to be passed on to an organism's offspring is carried out. This process is called meiosis. In animals, meiosis occurs in the testes and ovaries. In plants, meiosis occurs in the anthers and ovules. Meiosis is a type of nuclear division that gives rise to 4 reproductive cells. Gametes, each with half the chromosome number of the parent cell. 2 consecutive divisions occur. In the first, homologous chromosomes become paired and may exchange genetic material before they move away from each other into separate daughter nuclei. This is the actual reduction division because each of the 2 nuclei so formed contains only half of the original chromosome. The daughter nuclei then divides by mitosis and 4 haploid cells are produced. With these basic ideas in mind, let us examine in detail the events that take place during meiosis. Learning outcomes. After completing this lesson, student will be able to state the importance of having the chromosome number in the formation of gametes. Stages of the first meiotic division. Prophase 1. The chromosomes condense and become more visible. The nucleolus disappears. Centrioles, if present, arrange themselves at opposite sides of the nucleus and a spindle forms. Homologous chromosomes associate with each other, in that they lie side by side. As prophase 1 proceeds, the homologous chromosomes may become intimately coiled around each other and exchange genetic material. Metaphase 1. The nuclear envelope breaks down and the homologous chromosomes move to the equator of spindle. Homologous chromosomes are positioned so that the centromeres orientate towards opposite poles. Anaphase 1. Homologous chromosomes each make up a pair of chromatids joined at the centromere, moves towards opposite poles of the spindle. The sister chromatids also separate slightly from one another along their length, except at the centromere. The final stage of meiosis 1 is telophase. During telophase 1, nuclear envelopes and nucleoli form around the 2 new nuclei and the spindle breaks down. At the end of meiosis 1, a brief interphase occurs. During interphase, no genetic material, DNA, synthesis occurs. However, chromosomes recondense and the second meiotic division begins. The purpose of the second meiotic division is to separate the chromatids from one another. It should be noted that in some organisms, interphase does not occur at this point. Instead, the process moves directly from telophase 1 to prophase 2. During prophase 2, centrioles in the 2 daughter cells replicate and a new spindle is formed at right angles to the first one. Metaphase 2 consists of the chromosomes moving towards the equator of the spindle and orienting themselves towards opposite poles. By anaphase 2, chromatids part company and migrate to opposite poles of the cell. Note, the chromatids become the chromosomes of the daughter cells. During telophase 2, spindle disappears and the chromosomes become thread-like. New nucleoli and nuclear envelopes form. By the completion of meiosis, division of the cytoplasm occurs and 4 haploid cells result. The 4 haploid cells result from the original diploid parent cell. After completing this lesson, student will be able to explain the role of gametes in the transmission of inheritable genetic characteristics. Meiosis and genetic variation. Meiosis results in genetic variation by A. The shuffling of maternal and paternal chromosomes and crossing over at prophase 1. B. The formation of 4 genetically unidentical cells, telophase 2. And C. Sexual reproduction, which results in unique offspring since unique haploid gametes are fused. Importance of genetic diversity. Why is genetic diversity important? Genetic diversity is important for the survival of species. Environmental conditions continually change. As such, organisms with favorable combinations of genes from their parents will survive and pass on those favorable genes to their offspring when they reproduce. Summary. DNA replication. DNA replication occurs before meiosis 1 begins. Number of divisions, 2, each including prophase, metaphase, anaphase, and telophase. Association of homologous chromosomes. During prophase 1, the homologous chromosomes join along their length, forming groups of 4 chromatids. Number of daughter cells and genetic composition. 4. Each haploid N containing half as many chromosomes as the parent cell and genetically unidentical to the parent cell and to each other. Role in animal body. Produces gametes, reduces chromosome number by half, and introduces genetic variability among gametes. Meiosis and mitosis compared. Mitosis occurs in normal body cells, while meiosis occurs in the formation of gametes. In mitosis, the normal diploid number of chromosomes is passed on, while in meiosis, the chromosome number is halved to the haploid number. In mitosis, nuclei have identical genes. However, in meiosis, nuclei have genes mixed by crossing over. Mitosis produces identical cells, but meiosis provides variation, new genotype. Mitosis occurs for body growth and development, while meiosis occurs for fertilization to restore the diploid number. Mitosis ensures genetic continuity, but meiosis ensures variation. Phases of mitosis and meiosis compared. Prophase of meiosis 1 is longer than mitotic prophase. In metaphase 1, the 2 copies of each replicated chromosome pair at the metaphase plate, unlike mitosis. In anaphase 1, each chromosome moves to pole without chromatid separation. At the end of mitosis 1, the chromosome number has been reduced, but each chromosome still has 2 chromatids. The chromosomes are not replicated in the interphase between meiosis I and meiosis II. The chromatids finally separate in anaphase II. The final result is 4 haploid gametes, each with half the number of chromosomes present in the diploid cells. Now that we have outlined the process of meiosis, you should now be able to complete this past paper question on the Csec 2007 biology paper 03.